Okay, thank you, Dr. Gloria Conover, for presenting for Hear from Peers. Welcome. Dr. Gloria Conover is an instructional assistant professor in the Department of Medical Education. She earned her PhD in molecular biology and microbiology from Tufts University and completed postdoctoral training in cell biology at the University of Arizona. She was recruited to her first faculty position at Texas A&M University in 2008. Since 2020, Dr. Conover has taught research-focused courses for pre-clerkship and clerkship medical students at A&M Medicine. In this role, she developed innovative electives, MEID 820 and MHUM 823 for third and fourth year medical students and serves as the course director for a required first year core course called MEID 618. Through her teaching, she helps students appreciate the biological processes and molecular mechanism underlying disease. Her past research focused on protein structural abnormalities associated with familial heart disease and the discovery of effector proteins that facilitate Legionella pneumophila infection within lung macrophages. Hopefully, I spelled it correctly. But Gloria, I will give you the floor for presenting.
Thank you, Armando, for kind words and for inviting me to this seminar series. So, hello, everybody. Today, I'm going to talk a little bit about my exploration to use AI in the classroom. And I'm going to specifically focus on two tools. So, the title of my talk is AI Assisted Learning, Science Lens on an Illness Script. All right. So, what is an illness script? So in medical school, we teach students to think about illnesses in order for them to get a diagnosis, right? So in order for them to get a differential diagnosis, they need to understand the patient's history. So in thinking about that process, it takes several years for students to master this competency. But then for the third year students, they already have acquired and seen patients in clinic. They have mastered the foundational concepts. So before the AI became very popular after in the last 2 years, I decided to spend this semester looking at how to improve my teaching. So I was through going to seminar series, I was acquainted with visualization tool to sort of organize concepts and ideas. So I actually started. So today's story, I'm going to tell you about one student case, and how we can integrate the primary literature, scientific literature, into the sort of the origin of the diseases. So to start, I decided to ask AI, how would I go about this, integrating science into medicine? So this is what I got back by using ChatGPT. So if you were to integrate for initial content, I didn't specify in my prompt sort of who is the person building this. So it gave me a very general output. So the AI tool told me, well, use one of the AI search engines to get the initial content, and these are the subtopics, and then employ a dedicated AI concept map, which is what the programs call it, mind maps. So okay, I thought, well, maybe that's a good idea. So this is sort of what the summary, and it sounded convincing. So I'm going to tell you sort of when I actually went and did the experiment with one of my students' cases. I actually found that it's generally okay what it's saying, but I actually changed some of the ideas, and I'll tell you why as we go along. So that is sort of the argument that the machine told me. And after looking at different visualization tools, I decided to really go deep into one called Miro. And then by attending the Tamu Library AI seminar series, I got acquainted with another tool called OpenRead, and I'll talk a little bit more about it as we go along. So these are the tools that today I'm gonna talk about in more detail. All right, so as I told you, I was gonna take one case of one of my students from third year medical students. So in this class that I teach in the last 5 years or so before AI, we had a student build a concept map. So they selected a disease that they help treat in their third year medical student rotations. They're called clerkship rotations in the hospital. So in this case, it's a pediatric patient. So we had the student through sequential milestones get what we call a concept map. So this student came and drew with color coded the different tests that were ordered in the clinic. It presented to the neurology clinic and then sort of organized without AI help, sort of what is going on with this patient. And they also had something very interesting. Since this was a child, the doctor that he worked with decided to do genetic testing. So they figured out that there was a gene that was associated with the symptoms that the patient was presented. And they came up with, well, in the final report, the phenotype or symptoms in this case sort of were associated with a syndrome called Wolfram-Hirschhorn syndrome. And here I listed sort of the major hallmarks of the syndrome that this patient presented. So just one of them that I'll talk a little bit more in my next slides, and we'll go more in depth. We're not gonna go into every symptom. Is so a face sort of morphology that they call Greed helmet, and we'll talk a little bit more. So here you see that it's pretty busy. The arrows are connecting. We don't know exactly how they're related between the boxes. There's a little bit about the color codes, and then the biology part is here in the orange. So the goal here is to figure out if we can use AI to sort of try to clarify a little bit of research direction in this rare disorder, or not so common disorder. All right, so a little bit of history, and this is quite it's biological, so it's a little bit more language specific to biology, but this syndrome is sort of, the gene was discovered in 1998, so it's been observed in the literature, in the case report literature. These children have these facial characteristics, and it can be caused by deletions, or deletions of these or mutations, different types of mutations in these three genes. And then in the biology literature, they made mice that have deleted one of these genes, and they die very young from heart defects. So that means that that is a pretty important gene for development, at least that is evolutionarily conserved. So the particular patient had a mutation in one of the family members of these genes, the number 2, and that stands for the different parts of the protein, but it also has 2 other ones, and it's one of the smallest. So what the takeaway from this is that this protein is an enzyme, So it has a very long name. But what it does is that it puts metal groups into the histone protein in a particular residue or amino acid. And these are important for protein transcription, so sort of how protein is expressed during development. So as genes get turned on, they make different parts of the body. So there was a defect in that enzyme's function. So that's sort of in big terms how we understand what would a mutation in this gene do to the origin of this disease. All right, so the patient itself, so this is sort of how biologists understand this. In the top, you have the gene, which is DNA, and these are the introns and the exons. So it has 24 exons, so that's the coding part. And the bottom is the protein. So this is the protein in the NSD2 mutation. And our particular patient had a mutation, so a disruption here. So it created a shorter version. So it mostly had all the parts of the protein, but it lacked a little bit of the end part. And that is reported in the literature. I checked it. So, and there are other patients in the vast literature that exhibit mutations in different parts of the protein. You can see they're listed here. So it's not the only case in the literature. So this is something that has been recognized since last 1998, sort of the features. All right, so how can I help my students understand this at a much more deep level so they can have more critical analysis of how to help their patients more? These are young patients that are going to live long lives, so they need to have treatment plans to improve their quality of life because different things can happen. So initially, what I'm thinking about in improving this course that I teach for third-year students is for students to start using AI, using a broad prompt. So throughout the literature, it is important to think how you give the order or the prompt to your AI engine. So here I gave some specificity in the prompt, and I wrote it here in blue text, and said sort of what the patient population and sort of and asked AI. And I used the Miro. So I was trying to test if the content generated by this visualization tool was actually correct. So when I gave this prompt, this is what I get out. So it's pretty general, but it's overall is correct. So I mentioned previously the facial features, so that Greek warrior helmet, and I'll show you more later. And then there's other abnormalities, neurological and muscle abnormality and developmental abnormalities. Then it also, for medical students, it's very important to have the first step is how the patient describes, in this case, the parents describe sort of how did the disease manifest. But for a student learning how to deal with the disorder, they need to start thinking about the diagnosis and the management. So this is sort of what it takes longer later on in residency, but they get some introductory practice during medical school. So this Miro was able to give me a very general sort of how do you go in the clinic and do genetic testing. This has been known since the 1980s. This is more 1990s, and this is very, very old techniques. And of course, it's a multidisciplinary approach and sort of the management considerations. Another part of the idea is for the students to be aware of the different support and prognosis. So we're more interested in sort of how do you manage the disease with the information that you can deduce or infer from the primary scientific literature. All right, so that's fine, that's good. So then we can go to the next step, which is getting a more organized vision of how this particular patient case presented in clinic. So this is an actual patient. So this, I asked Miro and gave a little 1 paragraph description of the different symptoms that the physician collected. So this is what is an illness script, and you can see that it organized it fairly quickly with the groupings, which are pretty decent, to help really narrow down sort of what aspects can we follow, right? So here is sort of the behavioral autism and different, this is sort of the behavioral changes. So those are general things. So this is good for the physician and the medical student to review together and discuss. So this probably is a useful tool for that. So in the next step, sort of, we wanted to, this is sort of the second part of it. So it is a pretty large illness script. This is sort of the beginning. So it comes and groups them. Again, if we ask a physician if these are really good groupings, he probably, or she would probably say they're fine, they're okay for a big first step to understand. But I think it sort of tries to organize the areas to focus on for the next things. Again, so this is sort of what I wanted to show you. Since I showed you that there was some biological information in the workup that the physician did, we were able to relate that mutation to an actual described syndrome in the clinical literature. So for a student to start digging into this information, it's important to sort of recognize the symptoms and the signs in their patients. So here, Miro, in with careful prompting, was able to come up with the description of what a physician starts to look at. So here, sort of, if we narrow down, let's look at the genetic testing to look at the biology of it. And also, so here is sort of, you can verify the information by going to other sources and see that it is definitely a microarray analysis is important for that type of testing. The other thing that I mentioned before is these abnormalities in the face, and this gives you a more descriptive features of cleft lip, high nasal bridge, and prominent forehead. So when this is where I thought we actually need 2 tools, right? So I to my knowledge right now, Miro is not helping you, doesn't give you the information. So I took the TAMU Library's advice and looked into a tool called OpenRead and put just a very basic prompt at the beginning. What is the known normal function of the NSD2 gene? And OAT, it has a nice feature because it actually tells you the sources of where it gets the information. So here, as I told you before, and we knew before we use AI tools by looking at PubMed articles, that this is a transcriptional regulator that binds DNA. And so it was getting sort of more to what do we know about this gene. So here is where there was a report, and actually this is from a paper that shows that describe facial abnormalities of the patients harboring mutations in this gene. And it's an autosomal dominant inheritance pattern, meaning that if one parent has it, their children are in high chances of inheriting it. And even in the articles, they even say that they can do a composite face of the patients and group them by the location of the mutation. So this gives you a little more detail on what is happening, and it helps the physician get an idea if they're going to practice neurology, for example, sort of how to make diagnosis in the patients before the genetic data comes in. So as a sort of pause here, I want you to appreciate that we need to use multiple tools in order for us to teach. And sort of and this is sort of an example. So I'm going to give you another example of this. Is sort of, okay, in the literature, what is the biological basis according to preclinical animal models of the growth and facial defects in the face in this particular syndrome. So as you go through it, and you click and read the original publications, you see that a lot of work has been done, so they can actually map in through the medical imaging exactly sort of the range of defects that can happen in the nose. For example, here, and this is the citation. This is even very much before AI, but this is available. But and also from biological laboratories. This is a Xenopus labies, which is frog. This is one of the prime models for developmental biology. They can see sort of the order of the genes on how they form and make these facial abnormalities. So as you can see here are examples for the medical student to realize where this information come from, and it can help them figure out, arrive to that clinical diagnosis. So after the students appreciate that biology, they can even dig deeper. So when you can use Miro, going back to Miro, you can ask it, okay, so what are the mechanisms associated with defects in the amount of this gene, right? That's the term haploinsufficiency. So here is sort of where the student gets the knowledge that they acquired in undergrad and first year of medical school, and sort of starts thinking about how they want to focus their effort in their research. So this actually, since it's a gene that is early in development, it's affecting multiple signaling pathways in different parts. It's even implicated in cancer potential and other neurodevelopmental disorders and metabolic disorders. So it sort of gives a preview of what can happen to this patient. And it also sort of gives sort of actionable routes that maybe would be available in the future for potential treatment when they're adults or older people, because medical innovation is very fast, and these treatments may become available. So for the teaching purposes, so you can look at, and this is the last example I'm going to give you, is sort of, so we can look at signaling pathways and the methylation, since this is an enzyme. So when I go back to Miro and ask a prompt that asks for sort of give me the mechanisms related to DNA repair, and add the referee publications, and I even give specific author and year for this gene, what I see is I got this, right? So I got a name and a year, C for DNA damage response. But when I went and verified the old-fashioned way, which is pumping PubMed, you actually didn't get any searches. So that gave me a little bit of cautionary here that the sources may not be, they're not verified by Miro. So maybe there's some hallucination going on. And again, when I did change the words to just the gene and the last name, nothing came up. So that is a word of caution to rely on Miro with information that hasn't been vetted. However, when I went to OpenRead and asked for a similar prompt, I got sort of papers that I checked and read that gave me more information on what I was looking at. So this is a mouse diagram of the different cell groups that are developed at different times during development. And this is sort of what happens in the signaling, the Wnt signaling dysregulation. And there's a problem with the migration of this particular neural crest that is regulated by the enzyme, which is the enzyme that the patient had a shorter version of. So here you have a diagram that indicates that if you have abnormal migration, you get uncoordinated migration. So you have these malformed cranial features that are very important for diagnosis in clinic. And also you get other issues that can result in microcephaly. So that's a smaller head size. And here is, since this is not the only gene that affects this pathway, it also can affect the dosage, meaning that the amount of genes that are being expressed can affect other parts of development. So here the student gets a much more comprehensive understanding of what's going on behind that disease, and it can be in a good way to really monitor this patient over their lifetime. All right, so now to wrap up, I want to just briefly discuss some tips that I modify from what I learned in the TAMU libraries. So it's important for in the class to tell your students that if you present concepts and things that you have used AI, to sort of get them to test the technology and to see the drawbacks that it has, and have an open discussion about how can this tool help us, but also how you can have to be aware of its pitfalls or weaknesses. So it's important for to read sort of any tool. So it's just have the conversation with the students about sort of what type of information you put into the AI, or if you have to remove certain personal information, of course you do, to comply with the laws. And it's an ongoing process on how to integrate these activities into our assignments. So in summary, sort of what the takeaway here is that AI is sort of a tool, but human creativity and sort of the thinking process is not replaced by the machine. And it's super important to fact check those hallucinations that can happen. So from my point of view, these are my take-home messages. So I think mind maps are tools that are good for organizing concepts, especially when you have so many, and figuring out what part of it do you want to chase for research questions. And it's important to integrate the knowledge from the beginning of the class. But it's important to ensure that is accurate and relevant. For disease, to understand disease pathology, the AI can be useful, but it's important to emphasize that the primary literature needs to be reviewed and read carefully, because there's also information that the AI can extract incorrectly. And in my point of view, this should be done before using Miro visualization, at least at this time, because as I showed you in my presentation, there was some inaccuracies. And again, to hone in the point that it's essential to really cross-reference those sources. And with that, I want to thank everybody for your time today. And I want to acknowledge my collaborators and just want to close with how are you going to engage with your AI in your classroom after today's seminar. Thank you very much.
All right. Thank you, Gloria, for presenting this. Let me stop the recording for now. Hmm.